Hello friends. Today I will explain you some basics of DNA electrophoresis. It is very much important to separate the large size DNA fragments so that it can be analyzed in different purposes. It may be for identification for other purposes. So it is been achieved by electrophoresis. What is electrophoresis? It is a process in which fragments of DNA get separated based on their charges. It can be seen that DNA consists of phosphate groups in their DNA nucleotides. So it causes the DNA to make negatively charged. So when DNA is negatively charged, it can get migrated from cathode to anode in electric field during electrophoresis. Mainly the mobility of macromolecules based on charge to mass ratios and their shapes. So because phosphate groups of every DNA nucleotide carries negative charge, so the charge to mass ratio of DNA molecule is almost the same, though the length of the DNA fragment varies. So the shapes of linear DNA molecules are also similar. So during electrophoretic separation, different sized DNA fragments through a series of pores of the supporting matrix, either agarose or polyacrylamide gel, travels, and based on their sizes they get separated. So it is obvious that smaller size molecules will migrate faster than that of the large size molecules. Large size molecules will also experience some friction and collision during their travel through a matrix. What is this supporting matrix? Supporting matrix is actually a physical support material for a separating material. It consists of either agarose or polyacrylamide for separation. The agarose is a linear polymer which is composed of alternative residues of D and L galactose. A gelatinized agarose forms three-dimensional sieve with pores from 50 to 200 nanometer in diameter. As you can see in this diagram, there are certain small pores been formed after dissolving agarose and later cooling it. When DNA fragments from the range of 50 to 20,000 base pairs in size can be well resolved in agarose gels. Along with it, there is polyacrylamide gel, which consists of a polyacrylamide as a matrix. It mainly used for separation of smaller fragments of DNA. It is 5 to 500 base pairs. Additionally, a single nucleotide difference in the length of DNA fragment can also be resolved with this type of gel. This polyacrylamide gel is not only used for DNA, but it can also be used for protein molecules. So here is the general structure for polyacrylamide gel. In this case, you can see there is again a cross-linking being formed in between acrylamide and bisacrylamide, causing to form the small pores. So this is regarding some basic principles of electrophoresis of DNA and some matrices. Thank you very much.